Let me introduce our special guest. We have with us Dr. Stephen Matheson, who's an associate professor of biology at Calvin College in Michigan and visiting scientist at the Van Andel Research Institute. He has recently been critiquing signature in the cell. He earned his PhD in neuroscience from the University of Arizona. Sitting next to him is a very distinguished scientist, Dr. Arthur Hunt. He's a professor of plant and soil sciences and the director of the graduate studies for plant physiology at the University of Kentucky. In his 20 years of teachings, his students have gone on to become tenured faculty, research leaders, and high-level scientists in the industry. He earned his PhD in biochemistry from Brandeis University. Please join me in welcoming Arthur Hunt. And I thought we could hear from Dr. Stephen Matheson first. Dr. Matheson.
Let me tell you what I'm not going to do tonight. I'm not going to play the creationism game. That's a stupid game. It's a dangerous game for Christians like me because I'm a creationist. I sort of have to be, don't I? I believe in one God, the Father, the Almighty, maker of heaven and earth, all things seen and unseen, and I'm pretty sure that makes me a creationist. So I also am not going to quibble about the definition of science. Dr. Meyer has to deal with that in the book at some point about all this stuff about is ID science or is it not science, at least for the reason that we don't have a hard and fast definition of what science is to start with. Is ID science? My answer to that is who cares? Is it true? Is it useful? There are some interesting questions right there. The way I like to say it is I think design is the question, but not necessarily the answer. In other words, I don't think we should automatically assume that if there's something that fulfills certain criteria, like Steve has outlined, that we should conclude design is the answer. I'm not saying it's wrong, but I'm saying that's not my instinct. What I'm saying, though, is design is real, and we want to understand it. It's the question, not the answer.
I agree wholeheartedly with what Steve just went through. To give you an idea of how I'm going to approach this, really what I'm interested in is clearing up things that confuse me that I find in the book, things which get to some of the big issues, the things which touch on some of the stuff that I work on.
Well, so let's put up slide number 1. It's a phylogenetic tree. I think they're laughing because they recognize this from Stephen Jay Gould's book and they understand what he was getting at. So near the end of the book, you discuss the work of Doug Axe on protein function and protein space, and I'm going to read a quote from page 495. Referring to Dr. Axe, he realized that if functional sequences were common enough for mutations to stumble upon them relatively easily within the time required for relevant evolutionary transitions, mutation and selection might be able to build otherwise extremely improbable structures in small incremental steps. I'll stop and say that sounds right to me. That is what a standard neo-Darwinian account would look like. On the other hand, if functional proteins are extremely rare within sequence space, such that mutations will not have a realistic chance of finding them in the available time, selection will have little or nothing to work on, undermining its ability to produce biological information. This is in the context of a prediction, the end of the book, one of the predictions of intelligent design, and I think easily one of the weakest aspects of this book is those predictions. They're just not very strong. But this quote confuses me. It seems to me that to undermine the stepwise model of evolution that most biologists hold, and that would include me, you would need to argue that functional proteins are isolated in sequence space, so much so that mutation and selection can't reach them from other places in sequence space.
That's exactly part of Axe's program, both that they're rare and that they're isolated.
But instead, here, what's discussed is the rarity of functional sequences. I want you to look at this tree with me. Now, this could be a phylogenetic tree of anything. These could be people, they could be proteins, they could be species. It doesn't actually matter. It is, in fact, a tree from a book about very interesting ideas about evolution by Stephen Jay Gould. So what standard evolutionary theory says is not so much anything about whether these endpoints here are rare or common or easy or hard. What evolutionary theory does propose is that they're linked, and that this point there is linked to this place here through common ancestry. And that's all that we say. So what evolutionary biologists predict is not anything about rarity so much. In fact, we're willing to say, yeah, you need to be able to get there from here. You're not allowed to just jump over there. That's where you might calculate some outrageously huge probability of everything flying together, and nobody proposes that. So Is the point that these points are isolated, or is it that they're rare because those aren't the same thing?
Great point. If I failed to use the word isolated in the predictions section, I should have. In the long essay that I've written on the Cambrian explosion with Paul Nelson, Paul Chen, I know we made that point strongly, and it was also expressed in the very controversial article I wrote that was published at the Smithsonian's Journal. I actually think the best thing to do would be to let Doug Axe represent himself on this, but let me do my best explanation of the significance of his work as I understand it. He's going to be speaking tomorrow, giving a little mini lecture after the Cambrian explosion film, addressing the question of why mutation and selection are not sufficient to produce the information, the new genes and proteins you would need to build Cambrian animals. And his point is precisely that the functional proteins are extremely rare in sequence space and that they are isolated. Here's a way that may keep our general audience with us for understanding the point about isolation. Once it was understood, post-Watson and Crick, that genes are sections of digital code, it became apparent to some of the first doubters of neo-Darwinism, for example, the scientists who convened at the Wistar Institute in 1966, many of them quantitative types from MIT, that there was a kind of obvious problem. If a gene is a section of something like digital code or something like a section of alphabetic text, and you begin to degrade it at random by mutation, or you begin to change it at random, it raises the question, is it more likely that you will degrade that initial function or add to it as mutation progresses? As you map sequence space in some of the ways that Axe and others have done, it becomes possible to say, well, how many mutational changes can be absorbed in this process before you begin to lose the kind of function that is necessary to keep a gene in operation? Here's an illustration. Imagine you've got a sentence at the top, and the question is, can you get from one sentence through, you start to degrade it, time and tide wait for no man, and then you've got a sentence from Stephen Jay Gould. By degrading the initial information, are you going to be able to pass through a series of functional intermediates and then get to another meaningful sentence? In the protein world, it requires a continuous series of functional folds. And that's what Dr. Axe is questioning in his work, that it's not structurally possible to get from one stable folding motif to another, and there are other labs that are finding the same thing. So you're correct to point out, if I didn't use the word isolated, that's a critical part. That's definitely a part of his regime of research, and you're correct to point that out. And I will let him explain in more detail why those transitions from stable folds to stable fold motifs are not credible, and why, therefore, the isolation is a key problem at that fundamental molecular level.
Well, I heard a few things in there that are pretty controversial. I am not a protein chemist, but it's important, I think, to understand that how we think of things moving through sequence space is incremental. And Steve is right. We're proposing that every single step has to be possible. I don't think that at every single step during protein evolution you have to have a perfect function. You can drag garbage along for a little while in evolution. You can get away with that. I think the point is that some of the experiments that are described, and I have not read anything recent by Dr. Axe, the mutations that are made are huge, and they're made all at once. And the funny thing is, that's not how we propose that this works. It's surely an open question about whether that whole tree can be navigated through function all the way through. I certainly can't prove to you it's the case, but I want the audience to think about what the proposal really is from an evolutionary standpoint, and it is not that a protein can sustain a horrendous mutational hit and try to morph itself into something new. That's not how we talk about it.
It is an open question, and that's exactly what he's testing. And one of his results was that multiple position changes inevitably degrade function before they find new function, that there's a functional abyss between the equivalent of those two sentences, time and tide, or something from Gould.
Doesn't it matter that he made them 10 at a time? That the changes were made 10 at a time?
I don't think that's his experimental method. I think it's a pretty rigorous piece of work. He's got a new piece out that lays this out and a number of the other problems at the level of protein chemistry for generating genetic information by mutation in this new journal, Biocomplexity, the new research journal of many folks who are involved in the ID movement.
Question from the internet. Do you believe, Dr. Meyer, that the inference to ID as a cause in any way exceeds the bounds of science and enters the world of metaphysics?
We are going to talk about the definition of science. I think that those distinctions between science and metaphysics are difficult to draw. They can be somewhat arbitrary, and it is therefore possible for a scientific theory also to have metaphysical or philosophical or religious or worldview implications. And I agree with Professor Matheson that the essential question about ID is not whether or not it's science, but whether or not it's true. And the way I structure the case as I do, I very intentionally make my case for intelligent design based upon the same method of reasoning that Darwin himself used. I model my case after these established methods of scientific reasoning. What follows from that is that if someone wants to say that the case for intelligent design is unscientific by definition, then we would have to also say that Darwin and other scientists who use those same methods are unscientific. Typically, attempts to demarcate science from philosophy or metaphysics or religion are made by reference to methodological considerations, the methods that scientists use that are different, allegedly, than those other fields. And if the case for ID is being made on the basis of a standard method of scientific reasoning that's used by other scientists who are agreed by convention to be scientists, then it becomes very hard to disqualify intelligent design as not being scientific. I've examined these various demarcation criteria that are proposed, and what I found is that invariably, if they are strong enough, if they're applied in a restrictive enough way to disqualify intelligent design, then they have the unintended consequence, when applied in the same way, of disqualifying, for example, Darwinian evolution. One quick example: ID isn't science because it invokes an unobservable entity or an unobservable cause. Well, but so does Darwinian evolution, theoretical physics. In science, we often infer unobservables from observables. Science is often indirectly inferential, so that argument would cut both ways, and that's why these attempts to demarcate between science and metaphysics typically fail.
Dr. Hunt, your question.
I work in the field of RNA processing in plants, and my obsession is something called polyadenylation. When I come across a book that deals with gene expression in eukaryotes, the first thing I do is pick it up and say, what do they say about polyadenylation?
Did I not mention it?
You know, it's not in the book. In some of the discussion you have in your book talking about information, highly probable, improbable sequences, one of the things you say is that high probability sequences like ABC have very little information, either carrying capacity or content. Well, the poly A tail has a lot less information, carrying capacity or content than ABC. The poly A tail has tremendous function in the cell. The poly A tail tells the messenger RNA to be translated. It's a signal or a cue to stabilize the RNA. It's a signal or a cue to move the RNA through the nuclear pores into the cytoplasm, where it can be translated and stabilized. If you're a bacteria, the poly A tail is a cue to throw me away. If you're a transcript coming from part of the junk DNA in a eukaryotic genome, it's a signal to throw me away. So these things and your statements about information content leave me with some confusion, because there's a disconnect between function and information content. This and other examples actually lead me to believe that there's really no correlation between information content, specified information content even, as you define in your book and has been defined in the intelligent design literature, and biological function. There's just not a disconnect. Maybe some cases you can measure something you think is information and it might have biological significance, but in many other cases there aren't.
It's clear that digitally encoded information is responsible for generating function, but it may not be responsible for generating all function. I think it would be with biological signaling and with other kinds of systems in the cell, you may have something like structural information that doesn't lend itself to the kind of analysis which is essentially syntactic. When we're applying information theoretic constructs to biology, they certainly apply to the digital encoding of information. We have Shannon information plus functional sequencing or sequence specificity, but that's not to deny that there are other critical elements of the cell that are also part of its overall functional integrity that may be difficult to capture with those mathematical constructs. So one of the things we're going to talk about in the wake of the film tomorrow is the fact that DNA is not responsible for all the information and structure that is required to build a body plan. DNA is necessary, but not sufficient. And we explore in the film the role of hierarchically organized forms of information, some of it structural, that is necessary to build a body plan. DNA codes for proteins, but proteins have to be arranged into other structures within the cell to characterize distinctive cell types. Cell types have to be arranged into tissues, tissues into organs, organs and tissues into body plans. And yet DNA alone does not control all those higher order forms of organization. So what does control that? We know where some of that structural information is, but not all of it. One of the things that we explore in the film is that the hierarchical nature of information and the necessity of these higher orders of structure, order, and information pose a very significant challenge to neo-Darwinism, because it wants to explain the origin of new biological form at the lowest level of that biological hierarchy. I'm big on DNA, but I know that it's not the whole show. The Darwinians, however, are wanting to say that it's mutations in DNA that get selected and therefore are the ultimate source of new form. But if DNA is only coding proteins and other things are necessary to organize proteins, cell types, tissues, and organs, then you could mutate DNA indefinitely without respect to probabilistic limits, time, and chance, and the mechanism of selection isn't going to get the job done.
I'm very, very skeptical that you can break the link between any of these orders of structure and DNA. You can't do it, because this means that you're saying that some of these levels of hierarchy don't involve protein structure and function. And I don't believe it.
No, we're not trying to break the link. We're making a distinction between what's necessary and what's sufficient. There are targets in membranes, for example, that are heritable, that are necessary, that provide necessary structural information.
And these targets are made of?
They're made of proteins.
Thank you.
Yeah, but the structure themselves, it's not entirely governed by DNA.
But are they governed by proteins?
Let me give you another example. The cytoskeleton has protein building blocks, but the building blocks are identical. The overall structure is not given by the building blocks. It's given by something else. And that structural information is important to the type of cell architecture that is heritable from generation to generation. You use the same building block, the protein itself is not wholly determining that three-dimensional architecture. And this is another area of active research, not just with ID people, but in developmental biology. We'll be talking about it more tomorrow, and we'd invite you to join. In any case, I affirm the basic point of your question, which is that we can't always cash out function in terms of digital code, but we do know that digital code produces necessary function.
From Michael Shermer, in the world of artifacts, we can infer that designed objects have a designer. Tool makers make tools, automobile makers make automobiles. When it comes to natural objects, such as DNA, we have no experience whatsoever of a DNA maker making DNA. How can you logically infer a designer outside the laws of nature? Snowflakes are designed objects, as we know. They are designed by the laws of nature, so it makes sense to assume that DNA is no different, a design object, just more complex. Even if you infer that DNA must have a designer, how do you infer that Yahweh was the designer? What experience do we have that Yahweh likes to make DNA? We have no experience whatsoever of a DNA maker
yes, exactly.
making DNA.
But we do have experience of a cause which produces the key feature of DNA, which is specified complexity or sequence specificity or digital code, and that causes intelligence. As for Yahweh, that's a bigger question, which is exploring the implications of the theory of intelligent design. The theory of intelligent design is based on scientific evidence and a standard method of scientific reasoning. It gets you to the conclusion that an intelligence must have played a role on the basis of what? Our uniform and repeated experience, that we know that intelligences, conscious rational agents, are the only known cause of that key feature that DNA possesses, which is specified complexity. It can't identify the designing intelligence by itself. I'm a theist. There are other lines of evidence, other lines of argument, which in conjunction with the evidence for design and biology point in a theistic direction. But I think that goes beyond the theory of intelligent design. And the fact that I happen to be a theist doesn't mean that the theory is unscientific. If you say that, you're confusing the possible implications of the theory with the foundation or basis of the theory.
Well, just to follow up on the first question, would you infer that intelligent design could be something else besides a supernatural force? Could it be a natural being, for instance, that might be 100 years, 100,000, a million years ahead of us that would be able to design such a thing.
The inference that I'm making is to intelligent simpliciter. So the identity is not established by that inference.
So it could be a natural designer then.
The term supernatural does not figure in our work. Okay, that's something that's we're usually accused of bringing the supernatural in. To follow the method that Darwin developed, we're making our inferences based on uniform and repeated experience. Now, I have other reasons for thinking myself that the designer of life is a transcendent intelligence, but that goes beyond the theory of intelligent design, which is more minimalist and rooted in this method that I explicated during the lecture.
Thank you, Bruce.
So going along with this theme of things designed, in several places in the book you make statements. The quote I've got here says, "Of course, electrical wiring diagrams and schematics of computer hardware and software provide even better example of functionally integrated systems. Oddly, we find the same kind of integrated functional relationships and coordination inside the cell." And so arguments like this are a large part of your inference to the best explanation. These things look like things which are designed, intelligent agents designed and put together.
The point is, we're looking for features that give you analytical traction in a cause and effect analysis. The jargon among philosophers of science who study this method that Darwin uses, you're analyzing causal powers, and you're looking for causes which are uniquely known to produce the effect in question. Sometimes things are like things. DNA may be a number of things, but what's of importance is the feature that is the same in DNA as in digital code, as in human language, which is the presence of specified complexity. And that feature I give a careful analytical definition to. And so there are arguments from analogy are intuitively plausible, but not as rigorous as an inference to the best explanation. And I think the functional integration, this is not central to my argument. My argument's mainly about information, and I do a lot more work on giving that a tight analytical definition. But in the section near the end of the book that you're talking about, I'm looking at some other features of biology that are suggestive of design that I think suggest some promising avenues of research, again, because they're features that we know from experience from one and only one kind of cause.
Right. So, but what is lacking in these arguments by analogy is examples that are well known in molecular and cellular biology that completely contradict the idea that, well, the cell, its information processing systems, the way circuits work, are like what we see in computers or in design things, in engineered systems or whatever. So a very big omission that you've made is not mentioned at all is the system that cells use to turn circuits off. So when you turn your computer off or on, what do? You push a button. You turn your cell phone off, you're going to push a button. You turn your car ignition off, you turn a key. The microprocessor in your computer, when circuits turn on and off, transistors flip. Well, this is not what happens in a cell. In a cell, if a gene expression circuit is going to be turned off, the cell reaches in and rips out the switch and throws it away. A perfectly good working switch thrown away. If you were running your computer like a cell runs, every time you type something, you'd be opening up and ripping out transistors and putting new ones in. All eukaryotic cells devote tremendous genetic resources to this system of control, of throwing away perfectly good components, not flipping switches, ripping things out and throwing them away. This is how cells work. What I'm referring to is the large set of proteins called E3 ubiquitin ligases, which function in almost every gene circuit that you can imagine. And to me, at least, this inherent and wide universal feature of gene circuitry in living cells just flies in the face of the idea that these things look designed because they look like something is engineered or designed.
You used a couple of interesting words just in making your point. One was control, which is an engineering concept. Mm-hmm. The second is circuitry. Yeah. The fact that the circuit is different in its operation does not undermine the idea that it's functioning as a circuit. And we know of only one known cause of circuits, and that is engineering. And this is why in the book I made it careful to develop the case as an inference to the best causal explanation rather than an argument from analogy. Design arguments that were formulated circa William Paley were analogical. And the problem with analogical arguments of all kinds, whether for design or not design, is that they don't give you any metric to assess the importance of things that are different as well as similar. They're based on similarities, but most systems have similarities as well as differences. By targeting features like specified complexity or irreducible complexity, which Behe zeroes in on, I think we can get analytical traction concerning those features. What's the best explanation of those? As for the things that are different, it's certainly plausible a designer could have made a circuitry that used a novel form of control, even one that doesn't appeal to our aesthetic sensibility.
It's plausible the designer could do anything at all.
But that's not the basis of the argument. That's the point I'm making. I'm not basing the argument on the things that may be different than things that we find in human design. I'm looking at features that are known to be produced by one and only one type of cause. So that's the difference between an analogical argument, which has the weakness, which I think you're correctly zeroing in on, and an inference to the best explanation, which is more narrowly focused and I think not subject to those kinds of critiques or challenges.
I'm a Reformed Christian, and I'm pretty sure you are too. I'm a biologist. I think a lot about this stuff. I'm pretty sure I know a whole lot more about molecular and cellular biology than you do. I've read the book. I think about this stuff a lot, and I've reached a really different conclusion than you have. And I was wondering, why do you think that is?
The implication of your question is that I, as a lowly philosopher of science, rather than a biologist, should have convinced you as a biologist, since you know so much more biology than I do. And since I haven't, it must be a deficiency in the evidence. And I don't think that is the reason. The question is baited to get me to say, well, it's to imply that it's an evidential deficiency in the book. I don't think that's the case. What I've noticed in the critiques that you've made of the book online is that you're raising some interesting technical points, but you've been basically nibbling around the edges. The central issue of information and the origin of information is not something you've addressed in your critiques. And so I'd challenge you to think more carefully about the logic of the case that I'm making there. I commend you, A, for reading the book. Most of my critics haven't.
Yeah, I've noticed that.
I commend you also for eschewing these dead-end debates about definition of science. People are different. Since we're both Christians, I'll share some in-house stuff. But I once sat at a meeting of young Earth creationists, old Earth creationists, and theistic evolutionists. And someone was trying to get them all together so that they would all, come to an agreement. And what I ended up talking about was the complexity of weighing evidence. And I used this inference to the best explanation model to try to create a framework of mutual understanding among these different parties. And by the end of the week, I realized this wasn't an intellectual issue. It was bad relationship skills, basically.
It is not the fact that I don't buy the argument means the argument is wrong. I'm not that arrogant. Come on, people. It's that old, tired cliche. Sometimes reasonable people can disagree. And so I don't find the argument convincing, but I think I know why. And the reason why is, and I just figured out tonight, that you said that we will reason backwards from what we know works, which is that intelligence makes codes. All agreed. And we'll reason back and say, therefore, this is the one explanation we know that can do this. I buy that. I get it. It's obvious. Intelligence always creates information. My view is a little different. It's everywhere I look and every time I look, if I just wait long enough, there is a natural and sometimes even materialistic explanation for things. Now, don't I have the right to say, I'm going to go ahead and extrapolate that back, like Steve's doing, because that's just kind of my preference. And so what I want all of us to agree on is that it's pointless to say, Steve, don't be stupid. Design doesn't explain what you want it to. Well, of course it does. How could it not?
You're comfortable waiting for another explanation.
I am.
Which, in a strict sense, concedes that the one I've offered is currently best, but that wasn't meant to be a gotcha line. It really wasn't. And that we have a different philosophy of science, which is where the locus of our disagreement probably lies and where we should continue to converse. You caught some nice things of a niggly nature in some of your blog posts that came just in time for me to correct them for the paperback edition.
Okay, that's the final comment, though, on that line of thinking is design will always be an excellent and irrefutable explanation. I just don't see how it couldn't be. Art is saying, well, it doesn't look design to me. He's right, and there's some stuff that goes on in a cell. I don't know how you get design out of that. But it's easy. You simply say, in fact, you do say this, let's wait. Maybe we're going to see there's a really good reason for why a cell builds proteins billions a day and puts them straight into the wood chipper. Maybe there's a good reason for that. You said that. There's nothing wrong with talking like that.
One other thing I'd just say about the inductive argument that you offer from our experience of materialistic explanations always panning out. Since Michael Shermer has sent a question remotely, I'll tell a story about one of our debates. We're writing back from college somewhere near St. Louis, the one where Churchill, in Fulton, Missouri, where he gave the Iron Curtain speech. And I asked Michael, he has this deconversion experience, a story that he always tells about how he was a Christian and became a materialist. And I said, well, what convinced you that a theistic worldview was untenable? And he said, well, it was because science had just explained so many things. He said, like just the structure of DNA. I said, yeah, but what about the origin of the information in DNA? And he said, well, okay, I don't want to fight you on that too much. And I said, well, what about the fine-tuning of the laws of physics and chemistry? And he says, well, there's the multiverse. And then I looked at him. This is after the debate when you're being candid. And he says, okay, that's kind of lame. And what about the origin of the universe at the very beginning of time, where talking has, solved the field equations of general relativity, and the curvature of space-time goes to a 0 point such that space is infinitely tight. How much matter can you put in zero spatial volume? Not much is the answer. That's not a promising avenue for a materialistic explanation. And then what about consciousness? And Michael saying, well, yeah, okay, those questions, I agree. Those are things, those are pretty fundamental questions. In other words, materialism has explained everything but the really interesting stuff. So,
I am a materialist on consciousness, by the way, but yeah.
So, well, good luck with that. Good luck with that. So, I think it's perfectly legitimate to bring to bear on our understanding of the natural world what we know from our introspective experience of another reality, which is there are minds. And I think the materialistic worldview and the materialistic approach to science writes out of reality that very fundamental aspect of it. I love science, and I think there's been a lot of progress made in analyzing things in terms of material cause and effect. There are other aspects of reality that we know from experience, and if we turn a blind eye to them, we may miss some things that are very important, especially about these fundamental questions of origins.
What do you think it would take to prove to the scientific community broader that intelligent design is true?
Thomas Kuhn said that scientific revolutions take place one retirement at a time. And the ID movement is growing. It's not going to be like a switch that's formed. Look at the editorial board of the new Biocomplexity Journal. The chief editor of that is the head of the bioengineering program at the Helsinki Institute of Technology. He's ID friendly. People like that aren't supposed to exist in the world. Some of the people that endorsed my book were leading British scientists. I didn't know they were friendly to ID. The movement's growing. So persuasion happens in lots of different ways, but I think some of the differences that Steve and I were talking about are fundamentally about the rules of science. What are you allowed to consider as a possible explanation? And I think because of the exquisite nano and information technology that is present inside the cell, and because of the increasing awareness of nanotechnology and information technology in our modern world, I think that the younger rising generation of scientists is surprisingly open to the kind of arguments we're making, and I think they're very interested in what we're going to do with intelligent design. Just as Steve said, I think correctly, he has every right to say, I want to hold out for a materialistic approach to this mystery of the origin of life. There are other scientists who are going to say, I think I've heard enough. I think I'm convinced by the basic structure of the argument that the ID people are making, and I want to take that now as a working hypothesis and generate some testable hypotheses and use ID as a heuristic to see what else I can find out about the world. The world ought to look different if ID is true, and so we're going to go and see if what we think it ought to look like is actually what it does look like, and we're going to do some experiments on that basis. You don't have to have absolute proof before you can begin to investigate the heuristic fruitfulness of a new scientific idea.
I'll just say one more thing because this is sticking at the top of my mind. So Stephen has mentioned several times the idea of specified complexity in DNA. And the complex I work on has a lot of proteins in it, and several of the subunits of this complex defy the notion that specified complexity, as measured and defined in the book, is a real factor in the functioning of the complex. So in other words, there is not a highly rigid, strict amino acid sequence requirement for the functioning of many or
poly A
most.
tails?
Not poly A tails, of the subunits. So I work on what people might call an irreducibly complex structure. My work and the work of others has shown that this complex can be put together without requiring massive amounts of specified complexity.
Yeah, fair enough.
It's the assembly of this complex really probably requires zero complex specified information. And I think there are probably many more examples in the protein chemistry literature and the molecular literature of proteins, complexes, systems that formally have zero complex specified information than any that have clearly been shown to possess complex specified information. It's my opinion that CSI is something that has never been shown in any protein.
I was with you up until the last statement.
So
I think there are a lot of structures in molecular and cell biology that could very well plausibly self-organize by chemical factors that are not rich in CSI. But proteins are typically very rich in CSI.
well, I'm talking about proteins.
I understood that you weren't, and I would accept. And what I would say is that just because there are things that can be explained by self-organization doesn't mean that there are not also things that can only be explained by intelligent design because of their information content. So I would acknowledge that there are things of the sort you're describing. If you're saying there are structures that play a functional role that don't have a lot of specificity or information content, and there are many in the cell that could, therefore, I would say, yeah, they'd be good candidates for explanation by reference to self-organization. We're not saying that everything gives evidence of discrete design in the history of life, and that's part of the research program of intelligent design, is to tease apart those things that can be explained by intelligent design, those that can be explained by chance and law in some combination, things that can be explained just by self-organization or law. So what intelligent design does is it adds another explanatory tool.
I don't see where design is needed to explain anything in protein structure and function.
Well, proteins require information to construct, and there we'd go to the mat to say, yeah, you are looking at evidence of intelligent design because of the informational requirement, as distinct from that other system you just described, which was not rich in CSI and could be self-assembled fairly readily.
Dr. Matheson, last word.
I'm baffled by the claim that genomes are information dense, or that they're characterized by efficient and well-organized packaging of information. You mentioned folders and subfolders and so forth. And the reason I'm baffled by that is not because I don't think there's information in genomes, but because genomes on this planet vary over, is it 4 orders of magnitude? Gigantic variation. It's the human genome, 3.2 billion base pairs. There's a protist, an amoeba, with a hilarious name. Its genus is Chaos, and its species is Chaos. And that genome is 200 times the size of the human genome. I'm really confused about how that works to say, I think the genome's got really tightly organized information, if you're not going to tell us which genome. Some of them are tiny, a fraction the size of the human genome, with many of the same genes. And then you've got these humongous bloated genomes from pufferfish. No, sorry, the big one is mud puppy, I think. Anyway, what's up with that?
What's up with that? Richard Sternberg is speaking tomorrow, and I just invite you or anyone else to hear him on that point. He's a genomicist. He was formerly at GeneBank and NIH, and this is his area of how the genome is organized to maximize storage density. And it's a big subject, so he's the expert. He'll be talking tomorrow, and I'm going to punt that one to him. But I think you're right. The different genomes have different organizational features.
I think it means there's 200 times more information to make an amoeba.
Well, except that we're now learning about alternative splicing, and that you make multiple genes. You can get lots of gene products out of the same genes. We clearly have more structural information than an amoeba, so we're getting that from somewhere. And it may not be just the primary sequences. It may be how the gene products are put together in many unique and interesting ways as the cell retrieves information that's stored on a needs basis to be concatenated with other gene products. So it's that alternative splicing that may account for those higher orders of informational complexity.
We better wrap up. If you want to continue some of this discussion, just show up tomorrow morning. Sign up for our conference tomorrow.